Going to discuss hemoglobin estimation, short exercise for hematology practicals. Hemoglobin. As all of you know, hemoglobin carries oxygen from the lungs to the tissues and carbon dioxide from tissues to the lungs. It is composed of heme and globin. Heme is made up of iron and protoporphyrin, and globin is made up of polypeptide chains. Coming to the structure of hemoglobin, the gene helps in the formation of globin chain is located on the chromosome 16. The gene that codes for the formation of beta globin chains is present on chromosome 11. In adults, a minor amount of hemoglobin A2 with alpha 2 beta 2 chains is also present and constitutes less than 3.5%. There are various hemoglobin variants: hemoglobin A, hemoglobin F, hemoglobin S. Hemoglobin A is seen in adults and hemoglobin F is seen in fetal life. During the embryonic and fetal life, different types of hemoglobins will predominate. Gover 1, Gover 2 and hemoglobin Portland are present in early embryonic life. After 8th week of development, these embryonic hemoglobins are replaced by fetal hemoglobin HbF consisting of alpha 2 and beta 2 chains. Alpha 2 and gamma 2 chains also. This remains the predominant hemoglobin until after birth and constitutes 50 to 90% of the total hemoglobin. After birth, its concentration decreases to less than 2% by 30 weeks of age. HbA is predominant hemoglobin then onwards. HbA2 and abnormal hemoglobins are hemoglobin S, hemoglobin C, hemoglobin D and hemoglobin E. Coming to the functions of hemoglobin, heme acts as to bind oxygen reversibly and carries oxygen to the tissues. It also facilitates the exchange of carbon dioxide between the lungs and tissues. So, the functions of hemoglobin are mainly, it is the primary medium of exchange of oxygen and carbon dioxide. Indications for hemoglobin estimation. It is used to determine the presence and severity of anemia, mild, moderate and severe. In India, less than 10 hemoglobin gram per deciliter is considered as anemia, and severe if it is less than 7 gram per deciliter. And hemoglobin estimation is useful for screening of polycythemia condition also, and it is also useful to assess the response to specific therapies in anemias, and it is useful for estimation of red cell indices and also selection of blood donors. For a blood donation, the person should have minimum 12.5 gram per deciliters of hemoglobin. Methods for estimation include calorimetric methods, gasometric methods, chemical methods and specific gravity method. Calorimetric methods includes visual methods and photoelectric methods. Color based. These are based on the color of the hemoglobin or a derivative of hemoglobin. And the physical method, it is based on the specific gravity. Chemical method, it is done based on the iron content of hemoglobin. Gasometric method, it is based on the oxygen combining capacity of the hemoglobin. Spectrophotometric method, it is based on the measurements using spectrophotometric devices. Colorimetric methods, color comparison is made between the standard and the test sample. Visual methods include Dahlquist method, Salic acid Hematin method, World Health Organization hemoglobin color scale, and photoelectric methods include Scion Meth Hemoglobin Method, Oxyhemoglobin Method, and Alkaline Hematin Method. Coming to Salic acid Hematin method, the principle is when the blood is mixed with an acid solution, the hemoglobin gets converted to a brown colored acid hematin. This is then diluted with water till the brown color matches with that of the brown glass standards of the Salic tube. Equipment: Salic hemoglobinometer consisting of Salic graduated hemoglobin tube which is marked in grams and percentage, and there is a comparators with a brown glass standard, and these comparators are consisting of pyruvic acid. Salic pipette or hemoglobin pipette which is marked with 20 microliters are useful to collect 0.02 ml blood. A small grass rod or stirrer, dropping pipette, and reagent n by 10 HCl and distilled water. This is the picture showing the apparatus that is used for hemoglobin estimation called as Salic hemoglobinometer, and there is a square tube called as Helix tube. So it also consists of n by 10 HCl also. Specimen: EDTA anticoagulated venous blood or blood obtained by skin puncture is used for the hemoglobin estimation. First you have to take n by 10 HCl into the Salic tube up to mark 2 and take blood sample in Salic pipette exactly up to 20 microliters. Now you have to add the blood sample to the acid solution and mix it using a glass stirrer and allow to stand for 10 minutes. Then add distilled water drop by drop till the color of the solution matches with that of the brown glass standard. Take the reading of the graduated tube at the lower meniscus, and it is measured in grams. Advantage: it is the simple bedside test. Reagents and apparatus are very cheap. Disadvantages with the Salic method is it takes 1 hour longer time, and perfect matching with the brown glass standards is not possible due to personal error. Source of light will influence the visual comparison of the color, and the color of the brown glass fades with time. Hemoglobin F is not converted to acid hematin in this Salic method, and therefore this method is not suitable for small infants. Carboxyhemoglobin, methemoglobin and sulfhemoglobin are not converted to acid hematin. Precautions: you have to wait for at least 10 minutes for formation of the acid hematin. Remove stirrer while adding distilled water, and also while taking the reading. Tap water cannot be used as the pH of the water may vary the accurate values, and you cannot obtain the accurate hemoglobin value of the sample. Hemoglobin is increased in physiological conditions and pathological conditions. Coming to the physiological conditions, it is increased in newborns, and there is a diurnal variation, and during exercise, and in emotional conditions, and at high altitudes. Pathological conditions include cyanotic heart disease, polycythemia vera, burns, and the hemoglobin is decreased in iron deficiency anemias and other anemias, and also in leukemias. Now coming to Scion Meth Hemoglobin Method. It is the method of choice for estimation of hemoglobin because all forms of the hemoglobin are converted to Scion Meth hemoglobin except the sulf hemoglobin. And the principle is it consists of ferrous potassium ferricyanide which converts into methemoglobin and ferric ion. So this is reacting with the potassium cyanide and formation of cyan meth hemoglobin. The absorption of the solution will be measured in a spectrophotometer at a wavelength of 540 nanometers with a photoelectric colorimeter yellow and green filter. This is the most accurate method for estimation of hemoglobin and it is recommended by the International Committee for Standardization in Hematology ICSH because in this except the sulfa hemoglobin all the other forms of hemoglobin are converted to cyan meth hemoglobin and this cyan meth hemoglobin is stable and it is a reliable standard which is available for hemoglobin estimation. Equipment: photoelectric colorimeter or spectrophotometer, a Salic pipette which is marked and pipette 5 ml and the reagents used are Drapkin's solution consisting of potassium ferricyanide 200 mg, potassium cyanide 50 mg, potassium dihydrogen phosphate 140 mg, non-ionic detergent 1 ml and distilled water 1 liter or 1000 ml and cyanomet hemoglobin standard solution with known hemoglobin values is used. And the equipment is as shown in the picture photoelectric colorimeter or spectrophotometer and Salic pipette and pipette 5 ml pipette or cuvette. Specimen EDTA sample anticoagulated venous blood or blood obtained from the skin puncture. First you have to take in a test tube 4 ml of the Drapkin solution and to it you have to add 20 microliters of blood. This makes the 1 in 201 dilution. Stop the tube, that is stopper the tube containing the solution and invert it several times for mixing. Then let the test sample stand at room temperature for at least 5 minutes. Read the absorbance of the test sample and standard solution using a spectrophotometer and yellow-green filter at 540 nanometers wavelength with a filter against the reagent black, that is Drapkin solution. Hemoglobin value is calculated from the formula or from the graph. Hemoglobin in grams per deciliter is equal to absorbance of the test sample into hemoglobin standard into 201 by absorbance of standard into 100. This is the picture or graph showing the hemoglobin values at the absorbance at 540 nanometers. Advantages with the Scion Meth hemoglobin method are, as I said earlier, all the forms of hemoglobin except the sulf hemoglobin are converted to Scion Meth hemoglobin HICN. There will not be any visual error as no color matching is required. Scion Meth hemoglobin solution is very stable and its color does not fade with time. So reading can be taken at a later time also. So it's not a urgent or it is not to be recorded immediately. You can take it leisurely. Absorbance may be measured soon after the dilution. A reliable and stable reference standard is available from the World Health Organization for direct comparison. Even though it is having many advantages, it is having few disadvantages. First one is diluted blood and stand for a period of time to ensure complete conversion of hemoglobin. Potassium cyanide is a poisonous substance, and that is why Drapkin's solution must never be pipetted by mouth. And the rate of conversion of blood containing carboxyhemoglobin is slowed considerably, prolonging the reaction time to 30 minutes and can overcome this problem. Abnormal plasma proteins causes turbidity when blood is diluted with Drapkin solution. A high leukocyte count also causes turbidity on dilution of blood. Centrifuging the diluted blood can help overcome this turbidity. Now coming to Dahlquist method. A series of graph colors said to correspond to hemoglobin values ranging from 10 to 100%. Blood obtained from a finger puncture and placed on the piece of absorbent paper, and the color is matched against the color on the chart, and the corresponding reading is taken. This is a cheap and simple method. Error is 20 to 50%. In colorimetric method or the Dahlquist method, it compares the color of the drop of blood drying on a piece of blotting paper to the reference standards of color papers available in the booklet. This is quick and easy, but it is inaccurate. This is a Dahlquist chart that is used for hemoglobin estimation. These are the comparators provided by WHO. Coming to Oxyhemoglobin method, in this the blood is mixed with weak ammonium solution and the absorbance is compared with the standard. It is rapid and simple, but no stable solution is available. WHO hemoglobin color scale, it was first discovered by the Scott and Lewis, and the principle is similar to the Dahlquist method. It is also rapid, simple, inexpensive but reliable. 1 gram per deciliter for diagnosis of anemia. Printed sets of colors are available corresponding to the hemoglobin values ranging from 4 to 14 grams per deciliter. This WHO hemoglobin color scale efficiency is greater than 90% in detecting anemias and 86% in classifying their grade. It is useful for screening the blood donors and also children and women in health programs and also in iron therapy. This is a picture showing the WHO hemoglobin color scale. You can observe the person is comparing the hemoglobin sample with the WHO scale provided. Normal ranges for adult males is 13 to 17 gram per deciliter. For non-pregnant females, it is 12 to 15 grams per deciliter, and for pregnant females, it is 11 to 14 grams per deciliter, and for children 6 to 12 years, it is 11.5 to 15.5 gram per deciliter, and for children up to 6 years from 6 months, it is 11 to 14 gram per deciliter, and for full term baby or at their birth, it is 13.6 to 19.6 gram per deciliter. Coming to alkaline Hematin method. In this method, in place of N/L HCl, which we are using in Salic acid Hematin method, here we are using N/10 NaOH. So the hemoglobin is converted to alkaline Hematin by addition of this N/10 NaOH. Here carboxyhemoglobin, methemoglobin, sulfohemoglobin are converted, but fetal hemoglobin is not converted. Hence, this method is used to detect the presence of fetal hemoglobin. So, in the exam Viva question, Which method is useful to detect the presence of fetal hemoglobin? The answer is alkaline Hematin method. Now coming to Hardens method, in this hemoglobin is exposed to carbon monoxide. As a result, carboxyhemoglobin is formed. This test is very accurate, but the carbon monoxide is dangerous and toxic, so it is not preferred in labs. Coming to Dare method, in this undiluted blood is spread in thick films between glass discs and seen for direct matching. And in Spencer's method, color of oxyhemoglobin is matched usually. And this is a picture showing the specific gravity method. Here the solution consists of copper sulfate with a specific gravity of 1.0530. Hemoglobin more than 12.5 gram per deciliter. If a drop is kept in this solution, it will sink. If the hemoglobin is less than 12.5 gram per deciliter, the blood drop will float in the copper sulfate solution. With this, mass screening of blood donors is done. As I said, a drop of blood is allowed to fall into the solution of copper sulfates of various specific gravities and markings whether drop rises to surface or sinks to the bottom. If the blood drop sinks, its specific gravity is greater than that of copper sulfate solution, and this is mostly used for screening of blood donors. Specific gravity method, it is a rough estimate and it is made from specific gravity of blood, and the technique is copper sulfate we are using, so it is a copper sulfate technique method, and it is used for mass screening for selecting the donors. Coming to chemical method, in this iron content of the hemoglobin is to be first estimated. Indirect hemoglobin is derived from 100 grams of hemoglobin containing 374 grams of iron. It is a time consuming method. This method is used to calibrate all other methods of hemoglobin estimation. This is the apparatus showing the gasometric method of hemoglobin estimation. Oxygen carrying capacity is measured by this apparatus. 1 gram of hemoglobin will carry 1.34 ml of oxygen. But this apparatus does not measure carboxyhemoglobin, sulfhemoglobin, meth hemoglobin. This is a time consuming and an expensive method. Result is 2% less than the other methods. Coming to automated blood count method, it is a modification of the Scion meth hemoglobin method. The chemicals used here is sodium lauryl sulfate, imidazole, sodium dodecyl sulfate. Measurements are made at various wavelengths depending on the final stable product form.